Hello, let's learn about microbiology of infections. What is microbiology? Microbiology is originated from the Greek word micros means which means small, and logis means which means study of life. It is study of unicellular single cell or multicellular cell colony or acellular lacking cells or microorganisms microbes. That is all what is called as microbiology. Microbes are everywhere. They have existed on this planet for over 3 billion years and are essential for humans to live. Now, that was about the microbiology. What do you understand by pathogen? Pathogens are microbes that cause disease, but not all microbes are pathogens. Many microbes are normal human flora inside the human body. There are 3 billion human cells and 100 trillion bacteria, protozoa, and fungal cells. When these microbes appear in parts of the body where they do not belong, they cause infections. Microbes are microscopic organisms. They are categorized according to the biological classifications. Organisms are also classified into 7 main categories according to kingdom, phylum, class, order, family, genus, and species. Many organisms are known by the family, genus, and species. For example, bacteria are named by the genus and species such as E. coli, Staph aureus, Microbial tuberculosis. Some family of bacteria are known pathogens in healthcare, such as Enterobacterus family. Also, microbes are classified. They are classified either as bacteria, viruses, fungi, parasites, and if you see parasites, the morphology is slender and slender organisms. Bacteria are very microscopic, often round or over or rod-shaped cells. Viruses are like globular in shapes, while fungi are filamentous in shapes, and prions are very microscopic structures round or oval. Bacteria are unicellular with no cells or single cells. They can be different shapes like cocci, bacilli, ovoid, spirals, or coma shape. They occur in different arrangements, but some of the bacteria are harmful and cause infections of human beings. Shapes of the bacteria can be cocci, coccobacilla, or spiral shape, as you can see from the picture. Identification is done visibly with ordinary light and microscopic examination. It is by staining the morphology becomes very clear. Examples of pathogens are MRSA or Neisseria meningitis. Opposing pathogens they do not cause disease, but in the human beings with the weakened immune system they cause infections and diseases. Treatment is of the bacterial infection. While viruses are acellular, they are able to replicate in the host cells only. They tend to be very smaller than the bacteria. They grow in cell lines and identified using fluorescent stains or enzyme-linked immunosorbents assays serology. Examples of viral pathogens are influenza virus, measles virus. While fungi are unicellular and multicellular, mostly they are not dangerous but can be very harmful to health. They can be cultured and examples include Candida and Aspergillus. Treatment is similar for parasites, they live on or in the host organism and get their food at the expense of the host. They are unicellular or multicellular. The microscopic and some are adult forms. The examples are malaria and cryptosporidiasis. Treatment is by antiparasites. Prions are defective proteins that misfold. There is some evidence of transmission due to failure to remove prions from a reusable diseases have been observed. Brain biopsy or autopsy are other special lab tests. Examples of prions include Creutzfeldt-Jakob disease. Treatment is by supportive therapy only. No treatment has been identified. How does this bacteria and fungi cause infection? Let us understand about bacteria. Very important parts of normal flora. However, when they appear in the part of the body where do not belong, they cause infection. For example, bacteria of the GI tract can cause urinary tract infections. Closed areas such as bladder, joints, and bloodstream are sterile areas. Any organism from the other part of the body, if they enter the sterile site, they cause. Now, what is infection? Infection occurs when a disease-causing organism called as pathogen enters and multiplies. And because different outcomes, they may either cause no infection or may be subclinical or may they cause clinical infection based on the immune status of the host. When the body is exposed to such pathogens, many different types of exposure can occur. It can occur by a prick or by needle. And after the exposure, there are many factors that can lead to infection. Factors. Subclinical infection occur when the pathogen are present in the host without causing signs and symptoms. That is a subclinical infection. Or colonization. Or clinical infection when they produce signs. Now, three things are necessary for the infection to occur. Well, places where the infectious agent live, that is surface of human skin, are necessary. That is, source is necessary. A susceptible person is necessary, and transmission route is necessary from which the germs are moved. Source is an infectious agent or germ and refers to a bacteria, virus, or other microbe. In healthcare settings, germs are found in many places. People are one source of germs, including patients, healthcare workers, visitors, and household members. People can be sick with symptoms of infection or colonized with germs. Germs are also found in the healthcare environment, such as dry surfaces, wet surfaces, including catheters, devices, dust, and a susceptible person is another one who is someone who is not vaccinated or immunosuppressed, and for the infection to occur, germs must enter the susceptible person's body and invade the tissues and multiply and cause some reactions. Certain medications used to treat the medical conditions such as antibiotics can increase the risk of some types of infection. For example, life-saving medical treatment procedures such as catheterization, intubations, and surgery increase the risk of infection. Recognizing the risk factors, prevention measures needs to be applied to prevent it. Evolution refers to the refers to the way the germs are moved. For example, germs do not move themselves. It depends on the contact route, droplet route, and airborne route. So through contact, the burns and boils and wound infection passes and transmits. Then through droplets by sprays and splashes, through inhalation, inoculation that like means of injuries. Now, by the contact route means that germs by spread by touch, by either by direct or indirect contact, such as the MRSA infection and VRE infection spread by contact route, either through medical equipment or through when hand hygiene is not followed. Sprays and splashes can also lead to the person coughing, sneezing droplets into the air, can lead to transmission of droplet infections like diphtheria, pertussis, meningitis. Inhalation of these germs in the air droplets can occur when they survive the air currents for longer time periods and over longer distances. For example, tuberculosis, measles, and swine flu and COVID-19. Sharp injuries can lead to infections such as HIV, HBV, and HCV, when needle pricks and needles. When the body detects such as an outside pathogen, it develops an infection because the immune system reacts to it, and the signs and symptoms depends upon the immune reaction with the pathogens. So it can be in the form of fever, pain, redness. This includes gram staining, gram positive testings, and the gram test of plays an art to perform. So based on the gram staining, the bacteria or the virus or the fungi can be identified from the gram staining. So if you see the gram staining result, it can be either positive or negative. The gram negative bacteria can be seen on gram staining, or gram positive cocci or gram positive bacteria can be seen. And based on the morphology of the gram staining morphology of the bacteria, they are identified. Diagnosis of infection, the confirmatory takes it requires a culture. The microorganisms from the specimen needs to be cultured on a suitable growth media. And the time it takes for the culture to grow depends upon the type of the bacteria. Microorganism looks different from each other when cultured. So based on the they are identified using manual methods or automatic methods. Then after the identification of the bacteria from the culture characteristics, sensitive testing is done by using different antimicrobials, either manual method or through broad dilution using automated methods or manual methods. The sensitive testing against different antibodies of a particular organism is done, which takes about 2 to 3 days for to be for depending on the type of antibiotics and the type of organisms, the treatment is initiated. Broad spectrum antibiotics cover a many kinds of bacteria, and this broad spectrum antibiotics include antibiotics such as chloramphenicol, tetracycline, suplox, and others. The antibiotics have limited against only certain types of bacteria. Therefore, it's narrow spectrum antibiotics. Now, antimicrobial resistance is of course when these organisms develop resistance are ineffective against or the antibodies are ineffective against particular organisms. So microorganisms develop the resistance and they can pass into the other microorganisms by ways of different channels such as that by conjugation, transduction, or transformation, and various development of resistance enzymes and genes and influx and efflux mechanisms that microorganisms develop resistance. Now, what is the effect of the antimicrobial resistance? Resistant microorganisms can place more of a burden on the healthcare system in form of increased morbidity and mortality. They can increase the patient's length of stay. They can increase the financial burden on the patient. How does resistance develop? First, then they develop resistance against the normal flora, and then it gets get transferred to the other bacteria. When microorganisms become resistant to more than one drug, they are called as multi-drug resistant organisms. Now, the issue the world facing is the increase in the prevalence of the multi-drug resistant organisms or MDR. Increasingly important to identify such MDROs, detected by laboratory tests, and employ to preventive and corrective measures for the prevention and control of such multi-drug resistant organisms infection. Spread of infection from a host to a reservoir and from the various portal of infection and modes of transmissions. It's called the makes up the chain of infection. So one has to prevent and control the infection. One has to break this chain of infection agent is susceptible host to portal of entry to portal of exit and the reservoir. It requires controlling this infection requires the breaking the chain requires uh, treatment of the underlying diseases, the recognition of the high-risk patients, aseptic techniques to be followed, care of the catheters need to be followed, wound care, hand hygiene, sterilization, disinfection, standard precautions, transmission-based precautions such as contact precautions, airborne precautions, droplet precautions. The portal of exit needs to be taken care of such as excisions and secretions. The reservoirs need to be cleaned such as people, equipment, and water. And rapid, accurate identification of the organism is very essential to prevent and control the infection. Some of the major roles played by the clinical microbiology laboratory in infection prevention includes surveillance, outbreak management, antimicrobial stewardship, deliberations of the infection control committee, and education. In each of these areas, the clinical microbiology laboratory faces new challenges for the prevention and control of hospital acquired infections. Microbiology laboratories play a key role in informing the appropriate usage of antibiotics, ensuring first and second line antibiotics are used whenever possible. The quality of the clinical diagnosis is still essential as it tests need to be interpreted in light of. The investigative setup includes steps include recognize the problem, establish the case definition, confirm the cases, complete the case finding, establish the background of the disease, characterize the outbreak, generate the hypothesis, case control study or code study needs to be employed, and initiate the control measures and ongoing surveillance, document efficacy of the control measures. So the take-home summary is this: although not all organisms are harmful, pathogens can spread through the healthcare environment. Through the chain of infection, we can see how a pathogen can cause infection from the reservoir to the portal of entry in the body. Microorganisms can become a drug resistant through the misuse and incorrect usage of antibiotics, which leads to the spread of antimicrobial resistance. Pathogens can become resistant to many drugs, which is called multi-drug resistant organisms. Microbiologists use the various methods like gram staining, culture, and sensitivity testing to characterize the pathogens and help the clinicians make the appropriate treatment decisions. Clinical microbiology laboratory plays a very important role in infection prevention control and antimicrobial. Thank you all of you for your kind attention.